Good morning, everyone. Today we will discuss another topic of the chapter Mineral Nutrition. The name of the topic is Nitrogen Metabolism. The main objectives of this video will be to know what is metabolism of nitrogen, what is nitrogen cycle and what are its various steps, biological nitrogen fixation, what is the importance of enzyme nitrogenase, and what is the fate of ammonium ions. Nitrogen is an essential element of all forms of life. It is a constituent of proteins and nucleic acids. It limits the supply of food available in ecosystem more than any other plant nutrient. The atmosphere contains 79% of nitrogen by volume, but in the molecular form it is never taken directly from atmosphere. So it needs to be fixed, combined with other elements such as hydrogen, oxygen, etc. before it can be utilized by organisms. The chief source of nitrogen for plants is nitrate in the soil. The atmospheric nitrogen is fixed symbiotically as well as asymbiotically by variety of microorganisms. Nitrogen cycle. It is a process of cycle of events by which free atmospheric nitrogen is converted into its various chemical forms. The nitrogen cycle consists of four important processes that includes nitrogen fixation, ammonification, nitrification and denitrification. Nitrogen fixation. In this process, atmospheric nitrogen is fixed which is used by the plants. In this step, the molecular nitrogen is converted into inorganic nitrogenous compounds like nitrate, nitrites and ammonia. Nitrogen fixation is of two types, physical nitrogen fixation and biological nitrogen fixation. Physical nitrogen fixation. It is the conversion of molecular nitrogen to inorganic nitrogenous compounds without the involvement of microorganisms. Fixation occurs in nature by lightning and ultraviolet radiations by which nitrogen gets converted into its oxides on combining with oxygen. These oxides also get generated by forest fires, industrial combustions and by some power generating stations. You can see here molecular nitrogen combines with oxygen during electric discharge and results in the formation of nitrates. These nitrates combines with oxygen to form nitrates, hydrogen and water. Biological nitrogen fixation. The conversion of atmospheric nitrogen into inorganic nitrogenous compounds like nitrates, nitrites and ammonia through microorganisms are called biological nitrogen fixation. Only few microorganisms and certain prokaryotes are capable of fixing nitrogen. The prokaryotic organisms have a special enzyme called nitrogenase which can convert atmospheric nitrogen into nitrates and nitrites. These microorganisms can be of two types, asymbiotic or free living bacteria. These are the bacteria that are not present in close relationship with another bacteria or other living organisms in order to convert nitrogen into ammonia. For example, cyanobacteria, Amination, nostoc, etc. Symbiotic bacteria. These are the bacteria which lives in close association with other living organisms to convert atmospheric nitrogen into nitrates and nitrites. For example, rhizobium bacteria, which lives in close association with legumes, and mycorrhiza, which lives with vascular plants. Frankia and rhizobium both are unable to fix nitrogen when they are present as free living in soil. But when present in symbiotic association with plants, they become anaerobes and are able to fix atmospheric nitrogen. Ammonification. In next step, organic matter that includes dead remain of plants and animals are decomposed in order to produce ammonia by microorganisms like actinomycetes, clostridium, etc. Out of the ammonia produced, some of the ammonia gets volatilized and re-enters the atmosphere, while most of it undergoes the process of nitrification by soil bacteria. Nitrification is the process by which ammonia is converted into nitrite and then into nitrate. Ammonia produced by the degradation of manures may not be directly available to the plant. So it is first oxidized to nitrite by the soil bacterium like Nitrosomonas and Nitrococcus, and this nitrite is further oxidized to form nitrate by the soil bacterium Nitrobacter. The bacteria which helps in the process of nitrification are collectively called as chemoautotrophs. The nitrate thus produced is absorbed by the plant and then is transported to the leaves where it gets reduced to ammonia that finally forms amino group of amino acids. Denitrification. It is a process in which nitrate present in the soil is reduced back to free nitrogen. The process of denitrification is carried out by denitrifying bacteria like thiobacillus, denitrifications, pseudomonas, etc. Denitrifying bacteria utilize nitrates and nitrite ions as electron acceptor in place of oxygen. Nitrogen. In this picture of nitrogen cycle, it is clearly visible that nitrogen is first fixed into ammonia by two processes, either through biological process, that is through microorganism, or through lightning. This ammonia is further converted into nitrates and nitrites by the process of nitrification. This nitrification results in nitrates, that nitrates and nitrites are absorbed by plants, which further decomposes to form ammonia. These nitrates and nitrites are ultimately converted back into atmospheric nitrogen by the process of denitrification. Process of symbiotic biological nitrogen fixation. The process of symbiosis involves two living organisms that are living in close association with each other and both are getting benefit. Several types of biological nitrogen fixing bacteria associations are known. The most familiar one is the relationship of rhizobium with the roots of leguminous plants like garden pea, sweet pea, broad beans, etc. They get associated mostly on the root hairs cells. The root hairs curls and bacteria invade it. An infection thread is produced carrying the bacteria into the cortex where they initiate root nodule formation. Root nodules are small, irregular outgrowths of the roots. They are internally pinkish in color due to the presence of a special kind of pigment called leg hemoglobin. Symbiotic nitrogen fixing bacteria are also known to occur in the roots of certain non-leguminous plants like alnus, cycads, etc. The process of formation of nodule is a series of multiple interactions that takes place between rhizobium bacteria and the root system of leguminous plants. During the process, bacteria initially grows in the soil near the roots of higher plants. They are unable to fix nitrogen there, but after coming in contact with the roots of leguminous plants, they interact chemically and enter into roots through root hairs. Rhizobium multiply and colonize itself to the surrounding of the roots of host plant where it gets physically attached to the epidermal root hair cells. After attachment, the root hair gets curled up at the tip due to which bacteria invade the root hair. The enzymes from the bacteria degrade the part of root hair cell wall which produces a thread-like structure called infection thread. The bacteria invade the infection thread and reaches up to the inner cortex of the plant. The bacteria after reaching the cortex stimulates the initiation of formation of root nodules. Bacteria enlarges in size and becomes bacteroids, thus leaving the infection thread and enters the cell, that is cortical and pericycle cell to divide. This growth and division of pericycle and cortical cell leads to the formation of knob-like structure called mature root nodule. The nodule thus formed after division is finally responsible for the direct vascular connection with the host for the exchange of nutrients. Mature nodule, thus formed after a series of events and chemical changes, contains all the essential biochemical components, that is leg hemoglobin and enzyme nitrogenase. The enzyme nitrogenase catalyzes the conversion of free nitrogen into ammonium, that is the first stable product. The enzyme nitrogenase has two subunits, iron protein and molybdenum iron protein subunit. The iron protein component reacts with the ATP and reduces molybdenum iron protein, which then reduces the atmospheric nitrogen into the ammonia. Nitrogenase enzyme cannot function in anaerobic conditions as it is highly sensitive to molecular oxygen. Thus, for its activity, it requires anaerobic conditions. Hence, in order to protect this enzyme from oxygen, the nodule produces a substance known as leg hemoglobin. Nitrogenase is not active when these microbes live in aerobic conditions, that is free living, but becomes active or functional in anaerobic conditions during the event of nitrogen fixation. The nitrogen fixation takes place in the presence of enzyme nitrogenase only, which reduces nitrogen molecule into ammonia. Two atoms which makes up the nitrogen molecule are bound together by a strong triple bond. These three bonds are broken down in stepwise reaction to yield ammonia. Nitrogen fixation requires the following three components: a strong reducing agent, that is FAD, which is provided during the process of photosynthesis. It is needed to reduce the nitrogen molecule, that is to add hydrogen atoms to nitrogen molecule. Then is ATP molecule, that is needed as energy source. And the last one is enzyme nitrogenase, that is to catalyze the reaction. Ammonia formed as a result of nitrogen fixation is used by plants to form minor acids whose top amino acids are utilized for the process of synthesis of proteins and other organic substances. This picture indicates how nitrogenase enzyme is converting atmospheric nitrogen into the two molecules of ammonium ions. Enzyme nitrogenase combines with substrate, that is nitrogen, and undergoes three successive reductions to convert this atmospheric nitrogen into two molecules of ammonium ion. In this sequential process, in each step, two atoms of hydrogen are added to molecular nitrogen and one bond is broken between them. As after three successive steps, all the three bonds that are present in a molecule of nitrogen are broken and it results in the formation of two different molecules of ammonium. Roots of most higher plants absorb nitrogen from the soil in the form of nitrate. Nitrogen in this form, however, cannot be directly used by the plants and it must be reduced to ammonia before it is incorporated into the nitrogenous compounds of the plant. The process of nitrate reduction to ammonia is accomplished by two steps mediated by two specific enzymes that includes nitrate reductase and nitrite reductase. Nitrate reductase reduces the nitrates to nitrites. This reaction can take place in any part of the plant body. Nitrate reductase is a flavoprotein and contains molybdenum. Nitrite reductase reduces the nitrite ions to ammonium ions. Nitrite reductase does not require molybdenum and may contain copper and iron. Since ferredoxin is the direct source of electron, this reaction takes place in the leaves and the nitrite ions formed elsewhere are also transported to the leaf for reduction. Fate of ammonia. What happens to the ammonia generated after nitrogen fixation? It is protonated to form ammonium ion at physiological pH. Although plant can accumulate nitrate and ammonium ions are toxic to them. Thus, it is in turn used to synthesize amino acids in plants as follows. It includes two processes, reductive amination and transamination. Here, in case of reductive amination, ammonia reacts with alpha ketoglutaric acid to form glutamic acid in the presence of enzyme glutamate dehydrogenase. Transamination. Here, amino group of one amino acid is transferred to the keto group of a keto acid in the presence of the enzyme transaminase. Asparagine and glutamine, the two most important amides in plants, arises from two amino acids that includes aspartic acid and glutamic acid. As already discussed, the two most important amides found in plants are asparagine and glutamine. They are formed from two amino acids, aspartic acid and glutamic acid respectively. Both these amides are formed by replacing the hydroxyl part of the amino acid by amino radical. You can see here glutamic acid combines with ammonium ions to form glutamine, as well as aspartic acid combines with ammonium ions to form aspargine. Amides contain more nitrogen than amino acids and are structural parts of most proteins. Most amino acids. From amino acids to proteins. Proteins are built from amino acids. They are complex organic compounds containing carbon, hydrogen, oxygen, and nitrogen. A protein is a highly organized molecule made up of one or more polypeptide chains. A polypeptide chain consists of hundreds of amino acids in a specific sequence. The amino acids in a polypeptide chain are linked by peptide bonds. A peptide bond is formed when carboxyl group of one amino acid reacts with the amino group of other amino acids with the removal of one molecule of water. Given below is the formation of peptide by linking of two amino acids together by a peptide bond. Thank you everyone.